Hi, everyone. My name is Mike Sullivan. I'm the External Affairs Manager here at Skidaway Institute of Oceanography. Welcome to the September presentation of an Evening Skidaway. We were hoping we might have been able to be back in person this evening, but of course, you've probably been following the news and see that things aren't going exactly well enough for us to do that. For those of you who may have been working off the schedule we originally posted and put out back last winter, and we're expecting Dr. Clark Alexander to be here to talk about earthquakes. We apologize, but sometimes when you put out a schedule months and months in advance, some things come up. We did have a scheduling conflict, and Dr. Alexander will be presenting in October, on October 12th. This evening, we have Dr. Marc Frischer talking about a subject that I think is near and dear to the hearts of most of us, shrimp, and a condition called Black Gill that appears to be affecting the shrimp here in Georgia. Dr. Frischer is a full professor here at Skidaway Institute and the University of Georgia. He received his bachelor's degree in microbial genetics from Washington University, and then his doctorate from the University of South Florida in microbiology and molecular biology. He joined Skidaway Institute right at about 25 years ago in 1996. So Dr. Frischer, take it away.
Thank you, Mike, and also thank you, Dan, for hosting. Dan will be answering, sort of moderating questions at the end of this. Also, Wayne Aaron, who's the IT guy behind all of this magicry. And just a correction, Mike, I think that maybe not so near and dear to our hearts as near and dear to our stomachs as the shrimp. But thank you, everybody, for joining us tonight. I'm really excited to be here. I'd prefer, obviously, it be in person, but this is pretty good, too. So tonight, I'm going to be talking about shrimp black gill. It's a condition that has been affecting Georgia shrimp and Georgia shrimp fishery for quite some time. Actually, the same year I got here, turns out black gill got here as well. But before we get started, I just wanted to give maybe thought it would be helpful to give a little bit of background about sort of why this is sort of in my area of interest and primary research interest. And that's because I am a microbial ecologist, and I'm particularly interested in how the very, very smallest organisms on our planet interact with other organisms and the whole planet as well. And it turns out, as you'll see as we move forward in this discussion tonight, that black gill is one of those interactions. This black gill is caused by a microbe affecting a larger organism, shrimp. So here we go. All right, I imagine that many of you have probably seen this condition. Maybe you didn't know what it's called, but this is shrimp on the top here is a shrimp that has this condition, black gill, and you can see these are its gills, and the gills are melanized and they're darkened. That's a melanization. The same melanin that we have in our skins also occurs in other animals, and it's actually part of the immune system. So when the shrimp, anything really kind of goes wrong with the shrimp, and the shrimp turns on its non-specific immune system, oftentimes the consequence is melanizing or darkening the tissue. And you can see underneath it is a shrimp that does not have black gills, and usually it's the gill tissue is actually quite white looking, and you don't see any color there. So that's what we're calling black gill, and we're going to delve into a little bit about what's causing it and what effect it has, and is there anything we can do about it. All right, so this is a graph, a figure of both the performance of the fishery. So the y-axis on the left is what's been most affected by black gill, the fall harvest, which is the biggest part of the season. Most money is made in the fishery in the fall and fall landing. That goes through September, December. Sometimes we extend it to January, just past the new year. And this is the black lines with the circles in it. Those are landings, annual landings. So you can see back in 1985, 1990, the Georgia was landing about somewhere between 2.5 and 3 million pounds on average in the fall season. And sometime around 2000, that number dropped quite a lot. It's now somewhere more between 1 and 1.5, although there's quite a lot of fluctuation, as you see. The gray bars, that pertains to the axis on the right side, is the prevalence of black gill in the fall. And you can see that it was really not here until 1996 when I came, and I promise I did not bring it, hadn't even heard of it at that point. But in starting somewhere, the first observation was in 1996, official observation. Turns out when you go back and interview fishermen, they say, oh yeah, we've seen that, but it was never highly prevalent. And then about by 2000, it really was with us in pretty high prevalences, somewhere in the 20 to 40% on average. That doesn't mean a shrimper couldn't drag up a net and get 100% of their shrimp having black gill. In fact, last week, we got a sample just like that. 100% of the shrimp had black gill. And you can see that when there's a coincidentally or concurrently, when black gill came in, that's when we saw this really big decline in the fishery. Okay? But correlation is not causation. And so it's not clear that black gill is the cause. It's actually quite difficult to prove that black gill is the cause of this decline. Before we go any further, though, I want to make clear that shrimp and the shrimp fishery is still and remains by a large margin Georgia's most valuable fishery, part of its wild fisheries. Counts for maybe 2/3 at this point of the total economic value of the fishery. Okay. But in addition, as I said, many things are changing. Not only, you can't always just say that black gill is what causes decline. Many things are changing, and shrimp, just like almost any industry and any commodity now, is a global commodity. So we really can't think of it just in our backyard. We have to think much more globally about it. But in our backyard, our fishery itself, the number of people out there fishing, the number of boats licensed to trawl and catch shrimp commercially has been declining since 1980, when we have over 1,000 shrimpers out there. Of course, we closed the sounds, and that had a lot to do with changing the fishery. In 1979, that happened. So there's been a decline from that, clearly. But the fishery has declined. The number of trawlers out there have declined. The age of the average shrimper has increased. The age of the boats have increased. All of these things are happening. And of course, if you have less fishing, you might expect to be catching less shrimp. So that could be an explanation. Also, it's a global market now, especially about 80% of the shrimp consumed in the world and in the United States as well comes from farm-raised overseas shrimp. So most likely, if you go to the grocery store, especially if you buy frozen products, which are very good, that's not local shrimp. That's coming from somewhere usually very far away, from East Asia or South America. And the price of shrimp, that global commodity, fluctuates a lot. And of course, that changes at the dock here, how much a shrimper will get for his or her shrimp. So lots of things: insurance costs, fuel costs. So many factors go into this number of what's the landing fish. But for sure, the shrimpers, especially in the beginning, before we knew even what was causing black gill, the shrimpers really have put this together and said, black gill is the cause of our decline. We've got to do something about it. It's destroying us. And so I just wanted to, I hope this video works. Wanted to play just a little short video clip of shrimper, Captain George McKenzie, talking about this issue. This was recorded a couple of years ago by our own Mike Sullivan.
Well, I'm catching plenty of shrimp because there's nobody out there fishing on them but me. But it really don't mean nothing because the price of shrimp is down. And we know the fall shrimp's gonna have black gill in it. And that black gill gets in them. It kills them before they get to us. And, You'll go out there one day and you'll be tearing the shrimp up. 2 days later, they'll be gone. But they just die out. They get weak.
All right, so did you catch that? Captain McKenzie said they're there and then they're not there. Another fisherman told me it's like someone turned the shrimp spigot off. They're just gone. And he's blaming Black Gill. He says Black Gill makes them weak and they just die out and they're gone before they can catch them in the fall. So the first question that we've tried to address was we had no idea what was causing Black Gill. I had mentioned it before. Black Gill is part of the shrimp's generalized immune response to almost anything. So any kind of parasite can cause it, bacterial infection can cause it, some nutrient deficiencies can activate their immune system. And so it's not clear just because you have the symptoms of Black Gill, what's the cause of it. And we found out very quickly, this is one of the very first samples we ever looked at. And what you're looking now is under a microscope, a very thin slice of shrimp gill tissue that's been stained in a way that stains DNA rich and poor material. So these dark purple spots, all of those are what passes for a blood cell in a shrimp called a hematocyte. This is other tissue that doesn't have so much of the hematocytes in it. A hematocyte has the same function as our white blood cells and red blood cells combined. And then very quickly, you notice that the tips of the gills, okay, are these things, these round things with the dots in them. Those are definitely not shrimp, okay? They're clearly some kind of animal. It's a protist, a ciliate, we immediately surmise based on what it looks like. And you can see that in response to that ciliate being there, the shrimp has activated its immune system, including melanizing the tissue. And what they do is that melanized nodule, we call it, encapsulates it, surrounds the invading ciliate, and basically suffocates it because it creates this nodule that is gases can't pass through, including oxygen. So that really very effective in killing the ciliate. It's also not very good if you think about it for the gill, which is supposed to bring oxygen into the system and get them to these hematocytes. If you have a lot of your gill full of these melanized nodules that don't allow gases to be exchanged. So you can see that this is probably fairly damaging to the gill tissue. And as I said, we discovered, I put in quotes there because obviously it's there. We just identified it or saw it for the first time. Really, when we started this work back in early 2014, almost the first samples we looked at were just full of this. So you think, okay, we know what it is, but what is that ciliate? All we can tell it, it's something that's not supposed to be there. It's probably a ciliate, but what is it? It was clear, however, that this parasite is what's the cause of the symptoms that we see of Black Gill. So what this graph is showing is on the x-axis on the bottom here is the color of the shrimp. So it's sort of this inset photograph up here from shrimp that don't have any Black Gill to ones that have severely darkened black gills. And we correlated that with the number of ciliates that you can count on the microscope and the number of those melanized nodules that you can count. And you can see there's a very strong relationship, correlation coefficient, an R-squared value of 0.9, 0.95, very strong correlation, suggesting again that the number of nodules, the number of ciliates is what's causing that symptomatic Black Gill. But, and this was a big but, it took us 6 years more to figure out what that ciliate was. It turns out the reason why that is because like many, in fact, most microbes on this planet, we have no idea of what they are, especially ones that are not affecting humans, not affecting us. So we did some genetics and we worked out the whole life cycle of this organism, which is what you have to do to be able to name a new species. And we finally had enough information so that we were able to describe this species fully as a new species of a genus of apostome ciliate called Halophysa lanai. And we named it lanai after a ciliate biologist who just died just a year and a half ago, quite tragically in the field, named Dennis Flynn. So what was interesting about this is, well, it's very closely related. I don't know if you can see it, it's kind of small, I apologize for that. It's very closely related to another ciliate, Halophysa chatni, that we actually know quite a lot about. This was described in detail in the 1930s, and we know a lot about it, and it is definitely not a pathogen. Yet this one is very close genetically, just really teeny bit of difference, okay? But it has obviously some very large differences in that it is quite pathogenic. So we worked out the life cycle, and we think within the life cycle, understanding the life cycle gives us insight into why this causes black gill. So the life cycle all occurs except for the infectious phase, which is called a swimming tomite, free living swimming cell. Okay, the rest of the entire life cycle all takes place on the shrimp gill. Okay, and so there's a resting form called a phoront. Okay, and that phoront develops into a feeding form called a trophont. And because it's on the shrimp, it's feeding on living tissue. So it's eating living tissue in the shrimp. That's why the shrimp's immune system says, oh, you better not be here. We got to take care of you. And then it responds. If the trophon, if it gets enough to eat, it does well enough, it starts to divide. It starts to make inside many, many copies of these tomites this form, eventually bursts, and the cycle continues. And what these pictures are all these different life cycle forms and the way we worked out how that life cycle is. Now, if you compare that to chatni, you'll see it's quite different. So first of all, here's a scanning electron micrograph of Halophysa chatni and Halophysa lanai. Again, very genetically, very, very close, but clearly quite different from each other, especially in the life cycle, because what chatni does, it has the exact same forms, that life cycle. It has a phoront, and that phoront hangs out on the gill tissue, but it's just sitting there. It's not feeding. When it starts, when the shrimp molts, that's when the phoront turns into, morphs into this trophont, the feeding form. And the feeding form eats on all the nutrients and goo that's left over in the molt, not in the living tissue, but from the molt. And then it does its stuff. It starts to divide, make more tomites, and those releases them to go find another shrimp. So the difference is that in Halophysa lanai, the feeding happens in living tissue, and in Halophysa, very closely related, Halophysa chatni, feeding waits until the molt happens, and so it's not evoking the immune system. So we've also been looking around to see where where Halophysa lanai is and where it's affecting shrimp. It is not just here, although in our backyard, we're at pretty much epidemic proportions. We see these prevalences of 40, 50, 60, 70, 80%, very high, and that's why it's in red here. But we found shrimp that are infected with the same ciliate all the way up north through Chesapeake Bay. We haven't looked much further. We know it's not in Maine, where there's another ciliate that, in a different species of shrimp, that causes something similar. And it's throughout the Gulf of Mexico. And this is work that's done with 2014 through 2018. We found in all of these shrimp that are related to our shrimp here, they're called, the group of shrimp is these food shrimp, are called pinnate shrimp. We found them in all the species that are local here. Invasive species, the invasive tiger shrimp, Pinnaeus mododon, Indo-Pacific strain that's made its way into the Caribbean and here, and also raised in aquaculture globally. So it's highly prevalent, distributed, wide prevalence. And I only put this up here because we haven't been able to look in other places, but I suspect it has a global distribution. And it affects a very large group. It's not of shrimp. It's not just specific to the species that we have here. And I put this big star here is because while we've been doing this study And we've been making relationships and connections with other researchers and other fisheries and other groups in our region and beyond. The folks in the Gulf of Mexico started saying, we're seeing more and more. It looked like it's getting worse and worse. And last year, a group in Texas that we've been working with, and we have helped them develop their technology to study this, they reported prevalences of black gill even higher than we're seeing here. So just in the time that we've been sort of watching it, things have seemed to change. And keep that word in mind, change, because I think this is a moving target. And as we move through this evening, I hope that I'm going to at least seed in your mind the possibility that what's really behind the emergence of black gill is our changing climate. Again, some earlier data having to do with the seasonal distribution. It's got a very seasonal strong seasonal pattern to it. We typically see, this is some data from DNR, just looking at visible shrimp that have visible black gills, so they have the darkened gills. We generally, this is data from 1996 when we first started to see it through 2013. That's what the red line is. The bars are just in 2013, which was a terrible year for the industry. And you can see it really starts, rarely see it anytime before August. It persists through November, maybe December, and then goes away. And that's true up and down the coast. So this is from north to south here, Wausau, some of these major sound systems on our coast, basically the same pattern. It really peaks in September, October, again, in the middle of that important fall shrimp season. It sort of starts to come up in August, and sometimes it doesn't until September, at least during this period. When we began this project in 2014, one of the very first things we did was, instead of just looking at the symptoms, we knew we needed to look at the actual cause. And so we developed a PCR assay targeted to the DNA of this organism. This is an assay very similar to what the COVID assay is, PCR that we're all now so familiar with, except in the COVID, the SARS virus is an RNA virus, so PCR is targeted to RNA. Ciliates have DNA, so we targeted this PCR to the DNA of the organism, but it recognizes the ciliate now, the parasite, not just the symptoms. And what you see again in these gray bars, that's the visual prevalence of black gill. Again, beginning in August, peaking in September, October, and then declining and going away in the winter. But the presence of the ciliate actually starts much earlier. Usually April, May, we start to see it emerge. This is a anomalous number. It's hard to catch any shrimp at this point. So it was a very low number of shrimp. So probably we found one out of all the shrimp, but it shows up as looking significant. But really it goes away in the winter and it starts to come back April, May, that area. And it reaches much higher numbers than the numbers that actually have symptoms. So again, as a COVID analogy, you could have maybe a very mild case of COVID, and now, especially if you're vaccinated, and not even know you have it, but it's there, okay? But a smaller percentage get a really bad case. And so that's what seemed to be going on here with black gill. And I just want to, again, make the case here that this effect that we've seen in the performance of the fishery, that has really affected the fall shrimp. Okay. So the fall, what you see here is over time, the data from the fishery goes back to 1956. So it's really Georgia is really blessed with quite an amazing record of our fisheries. So again, it's the fall shrimp that are declining. If you look at the other parts of the season of the fish, we have two main fish species of shrimp. We have the brown shrimp and the white shrimp. The white shrimp have two seasons. They have two cohorts, two generations in a year. And the spring shrimp, the same species, that's been stable. No change there. No change in the brown shrimp that usually come in June, July. That's the main shrimp that we catch here in June and July. The shrimp that we start catching in August and we catch through December, January, that's the fall white shrimp, same species as spring, but a different generation. That's what's being affected. All right. So fast forward a little bit. So this is now the same kind of data I was sharing with you up until 2015. Something seems to have changed in 2016. In 2016, for the first time ever, we started seeing symptomatic black gill in June. And before that, thousands of samples looked at. We hadn't seen any before August, okay? And if you look at the black lines, again, that's the PCR data. Okay. That still's the same. Starts and comes in sometime between April and May, peaks in September, October, and declines and goes away. Okay. You can also see another change is that the prevalence of the symptomatic is much, much higher now, suggesting that we have much more intense infections than we had before. Much higher prevalence of the shrimp that are infected now have the bad part of this. They don't just have an asymptomatic infection anymore. Now they're symptomatic. So again, something seems to be changing. The other question this continues to strengthen is, where the heck does it go in the winter? We have a few possible answers to that question about where it goes in the winter. And it's an important question, I think, because if you're hoping or thinking about how you might manage this, one thing, if it goes away, maybe we can come up with some strategies to keep it from coming back. If it actually does leave the population, maybe we can keep it out. So one thing that we do know about this group of ciliates, the apistome ciliates, is that they're often associated with crustaceans, many, many species of crustaceans. And so that's the first place we looked, was with other crustaceans. And in fact, we found them. Now the numbers are pretty low here, so I wouldn't say this is an exhaustive list about where this could go, but we know at least that it can go into a number of different crabs and shrimp species, other crustaceans. Okay. Some we didn't find it in, but again, numbers are really low. Okay. One species that we did find it in a lot, we were able to, this is a relatively easy species to sample, the grass shrimp. Okay. And about a quarter of them were infected that we saw. But what's really interesting about that data, though, was when we saw those shrimp that were infected. And so this is a little bit of a complicated graph. I'll just sort of explain it a little bit. Again, this is the season from January through November. On the y-axis is the prevalence of black gill. This gray bar is the symptomatic, the prevalence of visible black gill in the white shrimp is in that gray bar. In the black bar, okay, that's the prevalence in the grass shrimp, okay? And the gray bar is the prevalence in the white shrimp, the commercial shrimp. And so again, you see our fall kind of prevalence in the white shrimp, and pretty much not before that and not in the winter. It's kind of opposite in the grass shrimp. We see them in the, we actually didn't look here in January, but in March, we see it pretty high and it declines that season and then starts to go back up again as black gill is sort of and white shrimp are declining. So these shrimp are everywhere. They're in the estuary. This is the shrimp that definitely the ingressing, the shrimp that are coming in, the white shrimp that are coming into their nursery grounds. They're sort of hatched and born offshore, and then they ride the tides and come back into the estuaries where there's enough food, and they grow very quickly there. And that's why we have such amazing shrimp here. They would encounter lots of grass shrimp at the time. At the time, they're actually occupying the same habitat. They're about the same size, you know, maybe 10 millimeters, maybe a little bit more. And they probably interact quite a lot. And perhaps this is the vector, this is how it comes back in. And very interesting, but if that's true, there's absolutely nothing we can do about it because we are not going to eradicate grass shrimp. And if we did, it would have terrible ecological consequences. But interesting. But it sort of shows you what we're kind of up against. Also from the shrimpers, they were very concerned that the problem started to occur when we closed the sounds. And when the sounds were open, they were, of course, fishing there, and they were sort of trawling on the bottom, and they sort of thought or surmised that maybe by doing that and sort of tilling up the sediments in the sound, they were somehow controlling this parasite because maybe it lived in the sediment. So we looked, and that was a quite plausible idea. It could be. We looked in the sediment and the water, however, not that many samples, but we've never found this ciliate sort of hanging out dormant in the sediments. And now that we know the life cycle very well of this organism, we know that it would not be able to persist outside of the shrimp. So it wouldn't be able to live and persist in the sediment. So that's probably not the place where the infection comes from. And I don't know if I've mentioned it to date in this evening yet, but I want to make sure that I say very clearly that although black gill is perhaps harmful to shrimp, it is not to humans. Okay, we actually don't know. It does seem like it's harmful. At least it causes those symptoms, but we know that it's not harmful to humans, and I can say that with a great deal of certainty. If any of you have eaten shrimp, local shrimp here in the past 25 years or so, you have undoubtedly eaten black gill. And I have heard no reports of anybody getting any kind of sickness or any kind of infection by this ciliate. All right. So let's ask that question, is it actually harmful to shrimp? And so for these sets of experiments, one of the kind of experiments we did are sort of physiological studies. So just like you would go to a cardiologist and they give you a stress test on a treadmill and they make you run and they make all sorts of physiological measurements that are really telling about how your heart is functioning and how your respiration system and your blood and all of that is working. We can put shrimp on these kind of stress tests on a treadmill. So this an aquatic treadmill, and we have shrimp that have black gill and not black gill, and we let them run on that treadmill. And first of all, I have to say, I had no idea what kind of athletes these shrimp are. They can run for hours and hours without stopping. Incredible. I think the record we had was one shrimp ran for 12 hours nonstop. Amazing. But if you compare the shrimp that didn't have visible black gill, this symptomatic black gill, to ones that did, the time to exhaustion was significantly reduced if you have black gill. So there is a burden of having all that melanized tissue. Now you're killing the ciliate, but you're also damaging your gills. And so that showed up in this kind of data really well. And I was hoping if these videos are working to be able to share with you a little bit of what that looks like, because it's I think it's kind of interesting. All right, so this is a shrimp. This is early in 20 minutes into the start of an experiment. And you can see this shrimp is just running along, not phased at all. In fact, it's going too slow. It has to speed up. And it just goes on. About halfway through a study, a little getting near the end, you can see this is a different shrimp, obviously. This looks like it has black gill, but it doesn't. And you can see it's trying to keep going, but it's kind of tired and exhausted. You can see it's still trying, but you can tell it's getting tired. And finally, an exhausted shrimp. That I think is the same shrimp. And you can see it's not even trying. And if this shrimp, we hypothesize that this shrimp got to a stage like this, there's no way it would evade a predator. It's all those moving and tail flicking and all those are really important strategies. In fact, the only way a shrimp can get away from a predator. And believe me, we as humans are not the only ones who like to eat shrimp. It seems like everything will eat a shrimp if it can catch it. And so that was the last set of these experiments we did with our colleagues in South Carolina DNR at the Marine Resources Institute, Research Institute. And they have these mesocosm systems. And we were able to get a number of different predators and get them all acclimated and starve them so they were quite hungry. And then in these big tanks in a very statistically robust manner, we added shrimp that had black gill. So this is sort of a cartoon of what these experiments look like, and an equal number that didn't have visible black gill. And then we put the predator in. We let them go for a few hours, and then we take the predator out and we count what's left. Okay. And at the end of it, we ran several trials of all of these. And at the end of the experiments, we would sacrifice the predators and look to see what they had in their guts too, so we can tell if they're eating shrimp that had black gill or not. We did that with 3 different predators. We did that with red drum or spot-tail bass, as they call them here, sea trout, and the blue crab. All of these predators, they operate differently, but they all like to eat shrimp. And in fact, we found that on average, all of these predators, when you combine it all, managed to eat shrimp that had black gill, that were affected by black gill, on average about 3 times more than shrimp that didn't have black gill. And so that's a little bit perhaps difficult to see on this graph, but what we have on this graph is the number of shrimp that don't have black gill, so asymptomatic shrimp versus symptomatic shrimp, the ones that did have black gill. If it was even, if it didn't matter, if you would randomly eat one or the other, just didn't matter, the number would all be on this 1-to-1 line. So you would have 5 shrimp that had black gill, and you would have 5 shrimp that didn't have black gill. And in fact, as you can see, the preponderance of the data from all these different experiments we did were above this 1-to-1 line, indicating that they preferred they were able to catch, they preferred because probably they were able to catch the shrimp that had black gill. So secondary mortality through predation is increased by black gill, but what about direct mortality? Does the ciliate kill shrimp? And this has been a really tricky question to ask, mainly because we don't, until very recently, we were not able to develop a control group of shrimp that we knew didn't have the parasite. All we could do was separate out, do they have visible versus not visible? But we know that a lot of times shrimp can be infected by the parasite, but not show the symptoms. So it's hard to disentangle this mortality. But what we do observe is very similar to what Captain McKenzie said, is it says, you're out there, you're fishing, you're tearing them up. He didn't say them, he said them. They're tearing them up, and then they're just gone. And that's kind of what we see in our experiments too. So in these experiments, we brought shrimp in, we put them in aquaria, had flowing seawater, we fed them, took away all the predators, very nice place for them to be. And we followed them through several months. And so what's in the gray line is this is where we see the prevalence of shrimp that have black gill. And this red line is where we see mortality. So this is calculated as weekly mortality. So what % of shrimp died in a week? Okay. And what we saw in this particular experiment, this was done in 2015, is that there was about a 2-week period right when black gill started to show up in August, and through the first or second week in September, where we saw mortality rates up to 80%, close to 80%. And that's very similar to what the fishermen are reporting, is that they're there and then they just disappear. Right. So that was quite, you can't say for sure definitively that this is called black gill, but that pattern of disappearing shrimp happens, and it's replicated in the lab under more controlled conditions, and it occurs coincidentally when black gill emerges. It's also very temperature dependent. So these are the same kind of experiments done in 2016 and 2018 as we got more sophisticated in our capabilities, our research capabilities. We could add temperature control. So in 2016, we added a cool treatment. And again, you saw it happen at a different time. Remember, this is the year that we first saw black gill emerging in June. So these experiments, this is happening in June. We saw this big, very short window of mortality, and that was reduced in cooler water. So when it's less stressful, when there's more oxygen in the water because it's colder, we had less mortality. And in 2018, we did the same kind of experiment, except that we saw this time The mortality was, again, there's a lot of interannual variability. That short mortality window happened actually in September, October, late September, early October. And in both the ambient temperature and the heated treatments, we had 100% mortality in a week or two, couple weeks of period. Whereas we saw that same mortality happening in the cooled treatments, but it was much reduced compared. And if you look at around surrounding those times, very little mortality is happening in these systems at all. So something's going on, and it, again, coincident with Black Gill. But again, we cannot say for sure. And this is a question we're really trying to still disentangle and get at, is what's the role of this parasite in this mortality? So what I hope that is sort of emerging from here is that there has been change going on. There are environmental parameters like temperature that can very much affect mortality and affect the presentation of Black Gill. And what is that? And we've looked at all sorts, trying to correlate with temperature and salinity and river discharge and chemicals and contamination in the environment, all sorts of things, too much to go into tonight. But the one thing that, and none of that correlate, and there are no relationships between any of those parameters that we looked at. Very frustrating to do that kind of work, but important to do it. The only thing that we found that was significant and replicable correlations with climate indices. Okay, the prevalence of Black Gill correlates with climate conditions. So what I have plotted here is the fall Black Gill prevalence. So that's again all of this fisheries data that we have. Okay, correlated with the ENSO index. So a positive ENSO index means that for us here in the Southeast that we typically have warmer, wetter winters. Okay, and negative ENSO indexes we have are cooler and drier winters. And you can see there's generally a positive correlation with the ENSO, meaning that when we have warmer, wetter winters, the following year, okay, so the following fall, that we get a higher prevalence of Black Gill. Kind of interesting. Okay. And so this is just taking a look at some of our data, Georgia water temperature data, which is kind of interesting. We collect this now quite relatively easily. It takes a lot of work to maintain the sensors, but we're collecting this data from a number of places on 15-minute increments. This is from the Sapelo Island Research Reserve, National Estuary Research Reserve on Sapelo Island in their Cabrita Creek sample. And what you can see, what I have plotted with this green line here is the summer temperatures. So you can see that's pretty much a flat line. There's not, there hadn't been a lot of change in our summer temperatures, okay? Nor has there been much change in fall or spring. But winters have been getting warmer. And that terrible year for shrimp that I mentioned is actually declared a fishery disaster for us. They're finally just now in 2013, they're finally just now distributing those disaster relief funds. Kind of strange, but that's this year that we had this particularly warm winter. But that's what's been changing for us here, is the winters have been getting warmer. Okay, and this correlates with what the performance of the fishery. So what I plotted here is in these two boxes, the one on the left here is pre-Black Gill, 1979 to 95, post-Black Gill, 1996 to 2017. And you can see this relationship with winter or January, in this case, water temperatures has really shifted. So pre-Black Gill period, landings actually increased with temperature. And in fact, we had some years that it got below 8 degrees, usually gets below 7 or 6 degrees Celsius. We actually kill shrimp, they freeze. But we had a relationship that the warmer the winter was, okay, the more shrimp we got in the fall. All right? That has really shifted now. Okay, first of all, you can see the numbers are quite really lower. These are much higher than they are here. We've already seen that data. There are less shrimp being landed in Georgia and South Carolina now. But the relationship between temperature and landings has also shifted. Now, as we've shifted towards warmer temperatures, the warmer the winters are, the less shrimp that we get. So temperature and climate really. The winter temperatures affecting the fall season the following year are affected by the winter temperature. And this is, again, this is now a little bit broader analysis of it. And this is just Georgia data. The past one was Georgia and South Carolina, but you see this pretty strong, significant relationship between the fall white shrimp landings and the winter water temperature, okay? And this turns out that perhaps this is part of the solution for us. The knowledge that this relationship exists and is maybe what's driving the declining fishery, perhaps at least if we can't cure the problem or get rid of black gill, at least we can adapt to it. We can adjust the fishery to it. And so a product like this might end up being quite useful. So what I've done here is I plotted all our shrimp landings against water temperatures, okay? And you can see that if your water temperatures are sort of in this average range from 12 to 15, we end up with kind of an average harvest, okay? If the winter temperatures are less than 12, but greater than 8, we have above average landings. And if they are greater than 15 degrees Celsius, we have a below average. And the way I think this is useful is because for any business, for any commodity, predictability equals stability. And what really happened, again, back to the example of the fishery disaster in 2013, was that everything was pointing towards it was going to be a banner year, or really a bumper. It was going to be a bumper crop because we saw all these shrimp coming in the spring and starting to grow up. They were thick in the estuaries that year. And globally, across overseas, a terrible, terrible new disease was rushing through all the shrimp farms and killing all the shrimp. And you probably don't remember, one of the first slides I showed was the global price of shrimp. The highest point on that value was 2013. So we had this sort of perfect conditions for the Georgia shrimpers to really make a lot of money. There were a lot of shrimp out there and the prices were really high because an unfortunate disaster befallen the shrimp farms, mostly in East Asia at the time. And so everybody who hadn't been shrimping for years got fixed up their boats. They bought new nets. Some of them bought new boats. They hired crews. We had crews, shrimp boats here from all over the country waiting for the season to open. And they went out and there were no shrimp. They lost money in fuel and they just were no shrimp that year. That was this year, 2013. Well below average, turned out a very warm winter. So we don't know if black gill was the cause, but either way, because they were predicting up here and they ended up down here, a lot of shrimpers went out of business. We lost a good chunk of our industry just in that year. Okay. So if we had some tool, some product like this that worked, that would be helpful for saying this is going to be a good year, bad year, we might prevent some of that kind of harm that can come to the industry. Any kind of nature-based industry. Agriculture is the same way. Okay. Of course, these are not the kind of models that we really like because they're based on hindcasting, not forecasting. They're based on conditions. The underlying assumption is that the conditions will stay the same. That relationship between temperature and fishery performance will stay the same. And obviously we know, looking at our own data, that that's not true. That can change. So really a lot of the research that is ongoing right now, and I'm showing a picture of Megan Tomichael, who's a PhD student working with colleagues Jeb Byers and Richard Hall in UGA in Athens in ecology. She's a Sea Grant fellow this year. Megan is working very diligently and hard, trying to really quantify some of the very mechanistic parts of the ciliate, how it gets transmitted, what controls whether you become symptomatic or not symptomatic, what's mortality, all of these at that fundamental level so that we can put it into not a sort of hindcasting model, but a mechanism-based model. And that will be, I think, much more useful and robust if we can manage to do it, and would open up all sorts of other possibilities for figuring out, are there other management strategies that we could do? Can we change the way we fish, the way the fishery operates to maximize the fishery and preserve its and maximize its sustainability? So that's ongoing work, and hopefully one of these years we'll get to hear Megan talk and tell us about some of her work. So what's the future of our fishery, our shrimp fishery here? And it is uncertain at this point. I really like this quote. Pat Gear, who used to be the chief of marine fisheries in Georgia, now moved up to Virginia. Maybe he's in Maryland, somewhere in the Chesapeake Bay. But he said this on one of the last things he said before he left the state to me. He said that shrimp won't go extinct, but the industry might if we're not careful. And I think that's really where we're at. Black gill is here. It's with us to stay. We don't know yet if that is really the cause of this decline. It's probably maybe one of many, many factors that's involved in what our fishery is doing. But it's not going away. And the real solution to the problem is to understand it and to manage for it and for the industry to adapt to it. And that's, I think, what the story is with climate change for everything, is that we're not maybe we'll be able to moderate some of the worst case scenarios of it, but it's here with us. We are dealing with it every day, everywhere. And I think this is another one of the things that we are dealing with, and an unpredictable and not predicted consequence of bigger changes that are happening on our planet. And with that, I think I'll stop and answer any questions that you might have. Thank you.
Thanks, Mark. I'll be the solo applause as the only person with a mic in the room.
Thank you, Dan. I appreciate it.
That was great. There's a couple of questions in the chat here, and apologies if you answered some of these earlier and later in the chat after they popped in. But are there other invertebrate hosts for the ciliate apart from the shrimp? Like, is it known whether the ciliate can survive outside of the host or through other hosts?
That's a very good question. We have found it in several other crustaceans. What it's doing in those crustaceans, we don't know. We haven't seen the kind of black gill that we see in the Penaeid shrimp in other crustaceans, but we do find it in other crustaceans. So it can at least hang out, maybe as that foront state, not being able to grow on living tissue of other crustaceans. But it's there. And there was a second part, Dan, to that question.
Yeah, whether it can survive outside of the host, like for any given period of time.
Yeah, all the data that we have at this point is no, but we don't know that very well. There is one part of its life cycle, if you remember, it's the swimming tomite phase, where it leaves the wherever it's been feeding and finds a new host. We don't have a good handle on how long it can persist that way. But that stage at least can persist for some time. I wouldn't expect it to be very long.
So I guess this is a related question that happened a little later. So is it possible or expected that the black gill will expand its range north as coastal waters continue to warm up the East Coast? Does that happen in other fisheries in other parts of the world where the pathogen is shifted over time?
Absolutely. I think of course the shrimp themselves are moving it, because the shrimp are moving north into colder climes. But we've seen it just in the time that the past 6 years that we've been really keeping our eye on this. We've seen it spread and become more of a problem all through the Gulf of Mexico, for example. And right now, the reason why our Pat Gear moved up north was because he got a better job, because they're talking about opening a fishery, shrimp fishery in Chesapeake Bay. So that fishery is actually moving north too. And of course, with it, it's bringing these parasites. So I think absolutely we can expect this black gill problem to spread and worsen as long as we continue to have these warming winters.
Ah, interesting. Here's a question about the treadmill. It's not from Senator Colburn, so it's from another academic here. So they're curious if the top speed of the shrimp is affected by the disease. So, like, have you ever turned the treadmill up all the way to see if their top speed is affected, or whether the black gill shrimp are just as twitchy as the uninfected ones?
That's a really good question. The top speed on that thing is equivalent of about 1 mile per hour, I think, something like that. And when we turned it to the top speed, the shrimp just kept up. We didn't run it that way because actually it started to break when we did that often. So it's a good question. We haven't explored kind of the performance of the shrimp, but we ran it at a slower speed just for a longer time. So I guess that's a good question. I like that question. Thank you. We haven't done that experiment. Maybe we should try it.
Interesting. And so relatedly, this is my thought too, is the higher mortality also associated with the higher temperatures during that part of the year? And could it be related to like the lower gas solubility, the lower oxygen solubility during that time of year? So like it's easier to stress them out when the temperature is warmer and it's harder for them to get oxygen because of the disease?
Right. I think all of those play together. So August, the time when we start to see that big mortality window, is when the parasite really seems to get going really well, and the shrimp are responding, and they respond, maybe they over respond. It's also the warmest time, and so the concentrations of dissolved oxygen, what they're breathing in the water, is lower, because the solubility of oxygen is related to temperature. The higher the temperature, the less oxygen the water will hold. So I think absolutely that's the stress of that system is increased at those times. It's really actually quite close here. It gets to be 30, 31 to 30, even 32 sometimes, the water temperature Celsius. And that's almost the limit, the physiological limit for this species. So when it gets like that, no matter what, it's stressful for shrimp. And everything is moving faster. The blue crabs, which are eating them too, fish, everything's moving faster and easier to catch them. And there's less oxygen there. And the parasite is probably liking the warmer temperatures too, and it's growing faster. All of those things is like a perfect storm that we think makes the mortality, probably increase mortality. It certainly increased temperature, if you have black gill in the population, increases mortality. Cooling the water decreases it.
That's interesting. Great. I have just about 8 o'clock, so we should probably end there.
Great. Well, thank you very much. I think is my email available, Dan? Is that made available?
I don't see it on unless you have a last slide showing the upcoming talks, which is sometimes part of the package.
I could switch to that. I'm not sure. Well, you can find. Right. Yeah, you can find my email on the Skidaway. If you go to the Skidaway website, www.skio.uga.edu, you'll look on people, and you'll find my name. Please, I'd be happy to answer any more questions that way. And again, I really appreciate everybody who sat through this virtually on YouTube. As Mike started off this evening, we really hope we can get back together soon.
Yeah, looking forward to see everyone in person. And if either way, there's another Evening Skidaway talk next month, so look out for the announcement for that.
Wait, I think I have that, if I can get to the right.
Yeah, I think it's the next final slide there. Yeah, exactly. So okay. Dr. Clark Alexander is presenting on Tuesday, October 12th, same time and place. Even if we are in person, we'll almost certainly be streaming it live online as well. So Clark's talk will be about earthquakes and tsunamis. So
and I think there's one more slide that Mike gave me.
Yeah, yes. And Dr. Jay Brandis will close out the year in November, the series in November. So yeah, we're looking forward to you. And then we'll have a new schedule up for 2022 sooner rather than later.
Hard to believe it's almost another year.
Nice seeing everyone.